Hi everybody, I welcome you all to this video lecture, Life Cycles of Viruses. The video lecture is being given by D. Rajashekar, lecturer in botany, Srimati Ganganeni Kalyani Government Degree College, Vinikonda, Guntur district. We all know that producing more and more number of cells or offspring is an important feature of a living organism. As we have learned, virus is the smallest living entity, though it is called a non-living organism outside the host. When it enters into the host, it can increase its number by replication process. That is why we have decided virus a living entity, but a complete parasite on the host. In this video lecture, you are going to learn how does virus complete its life cycle. So, the life cycle of virus has been well studied in bacteriophages, especially T-even type of bacteriophages, lambda-phages, etc. Actually, viruses complete their life cycle by two methods. One is lytic type of life cycle and another one is lysogenic type of life cycle. Whatever be the life cycle, the steps that are seen in two types are almost common. You can see in this slide the various steps that are involved in the life cycle of viruses. They are attachment, penetration, transcription, biosynthesis, maturation, and lysis. Before going through all these steps one after one, I would like to define what is lytic life cycle. Lytic type of life cycle was first studied in T-even phages by Max Delbruck in 1940. Here in this life cycle, virus kills the host, that is bacteria, after completion of life cycle. So at the end of life cycle, that host organism is killed so that all the viruses come out. That type of life cycle is called lytic life cycle. It was well studied in T-even type of bacteriophages such as T2, T4, T6, so on. First of all, how do viruses attack bacteria? Bacteriophage, we all know, it is a tadpole shaped structure with head, tail region. Tail is with base plates and 6 tail fibers. With the help of these tail fibers, bacteriophage or virus attaches the bacterial host. With the help of ionic bonds, the tail fibers of bacteriophage or virus attaches to the cell wall of bacteria at particular places. That is called attachment. After proper attachment is done, the bacteriophage or virus secretes an enzyme called lysozyme. This enzyme ruptures the bacterial cell wall at particular point and makes a way through which the bacteriophage or virus DNA material or genetic material enters into the bacterial cell. This is called penetration. For this, the contractile sheath around the tail of bacteriophage or virus helps a lot. Because of contraction and expansion movements of this tail sheath, bacteriophage gets a type of force by which the DNA material present in the head region gets injected into the bacteria or host forcefully. This is called penetration. Thus, in the bacteriophage, only genetic material got entry, whereas the capsid remains outside the host, as you see in the diagram. So the capsid which remains outside the bacteria host is known as ghost, since it is like a ghost. Thus entered DNA or genetic material of bacteriophage or virus immediately undergoes transcription process. That means by secreting nucleases from the virus or bacteriophage, the bacterial DNA is broken down into pieces so that virus gets control of all metabolic activities, enzyme systems of the host. Later on, this viral genome DNA undergoes transcription process and forms mRNA. This is called transcription process. Thus formed mRNA takes the help of ribosomes of host, that is bacteria. It prepares number of capsids by a process known as protein synthesis. After capsids are formed, the viral genome replicates and forms many number of DNA genomes. The so-called formation of more number of capsids as well as more number of DNA strands is called biosynthetic process. Later on, virus enters into the maturation phase. During this phase, the components of viruses get assemblage so that they can form complete virus particles called virions. This is called maturation phase. As these three steps such as transcription, biosynthesis and maturation take place in the host metabolic activities and all other activities are under control. So this is called eclipse stage for the host. And finally, that is lytic or lysis stage. Basing upon the number of virions which are formed inside the host, again these viruses secrete lysogenic enzyme which acts upon the bacterial cell wall and rupture the bacterial cell wall basing upon the number of virions, so that through these entry or rupture, all these virions or bacteriophages come out from the host, so that finally the host is killed. So the entire life cycle is over in just 18 to 20 minutes from the stage when viruses or bacteriophages attacked the bacteria. Thus, lytic cycle would be over. So in the lytic life cycle, actually it was described by Delbruck in T-even phases. In lytic life cycle, virus genome gets entered into the host, but it does not get integrated with the bacterial genome. It remains separate till the end of the life cycle. By taking the control of the machinery of bacteria or host, it replicates and increases its number. After formation of many number of viruses, that is nearly 150 to 200 bacteriophages or virions or viruses, by rupturing the cell wall of host, they all come out. So how much area of cell wall is damaged by the viruses to make out is called burst size. Thus, at the end of lytic life cycle, the host, that is bacteria, is killed. And lysogenic life cycle. This lysogenic life cycle was studied by Andre Loaf in lambdaphage, which generally attacks E. coli bacteria. E. coli is the host for this lambdaphage bacteriophage. In lysogenic life cycle, as of lytic life cycle, virus attacks bacteria by a process called attachment. Then by secreting lysozyme enzyme, it makes an entry on the cell wall of bacteria. Through that entry, viral genome gets entered into the host. that is called penetration. But later on, without being separated, the viral genome gets integrated with the bacterial genome. So as you see in the picture, as soon as the viral genome enters into the bacterial cell, it gets integrated with the bacterial genome, so that we can see the red colored structure as well as the green color structure. They are formed like a ring, and that is the genome of both virus as well as the bacteria. That is called prophage. That means viral genome got integrated with the bacterial genome. So along with the bacterial genome, viral genome will also be increased its number by the replication process carried out by the host organism. Thus, for some point of time or for particular time, both viral genome and bacterial genome being together gets replicated and give more and more copies, so that the host bacteria undergoes binary fission and form more number of bacteria. All these daughter bacterial cells are with this integrated genome of virus as well as the bacteria. Thus, lysogenic life cycle goes on. So it doesn't harm. It doesn't harm the bacteria or host. They maintain a type of friendship in order to get replicated together. Host is not immediately killed here, so it goes on for many generations. But when the host bacteria is exposed to certain chemicals, or by any artificial means or by natural means, or when they are exposed to ultraviolet rays that come from sunlight, so that automatically the bacterial host and the viral genome enters into lytic cycle. So far they have followed lysogenic type of nature. But when they are exposed once to these chemicals or physical rays, that is ultraviolet rays, automatically virus switches over to the lytic type of life cycle. So then the viral genome disassociates from the bacterial genome, and the rest of the process is more or less similar to that of the lytic type of life cycle. So lysogenic life cycle is integrated with the lytic type. That is the difference between the lytic life cycle and lysogenic life cycle. It was well studied in the lambda phase, which attacks E. coli bacteria. If you come across the major differences between the lytic and lysogenic life cycle, they are like this. In lytic life cycle, actually the virus kills the host immediately, that means soon after the life cycle. In case of lysogenic, the virus does not kill the host immediately. For some time, they maintain a good relation with the host. The host is not immediately killed. In lytic life cycle, as it is the virus is harmful for the host, the virus is called virulent phase, harmful phase. In lysogenic, as here the host is not immediately killed, the virus can be called as temperate phase or non-virulent phase. And the third difference between the lytic and lysogenic is, here viral genome does not get integrated with the bacterial genome. Hence, we cannot see prophage over there. So no prophage here. In lysogenic life cycle, both bacterial genome and viral genome get integrated, so that they form prophage. Prophage can be seen in case of lysogenic. In lytic life cycle, virus genome replicates independently without support of bacterial genome. It takes the control of bacteria enzyme system as well as other metabolic activities. So independently it replicates, so that it gives more number of capsids, similar DNA genomes, etc. But here lysogenic, here RNA genome gets integrated with the bacterial genome, and it depends upon the bacterial genome to get replicated along with the host genome. And another thing is, here in lytic life cycle, virus completely uses the host machinery, host enzymes metabolic activities. But lysogenic, the viruses do not use the host completely. Just they remain, very close to the bacterial genome. Both, actually go through a type of positive relation. That is why here viruses do not use the host completely. The best example for lytic life cycle is T-even type of phages, T2, T4, T6, so on and so forth. In lysogenic, that is lambda phase. So lytic life cycle remains as it is from first to the last step. It remains lytic. In lysogenic type life cycle, in the beginning, it follows the lysogenic type of life cycle. When the virus or the host bacteria gets exposed to your ultraviolet rays or some chemicals, immediately the lysogenic type of viruses change to lytic type, so that by killing the host they come out. So these are some of the major differences between the lytic and lysogenic life cycles. I hope you have understood this topic very well. So we'll meet again in another video lecture. Till then, bye. Have a nice day. Thank you.